Everyone, today we are exploring Chapter 4, which covers endocrine responses to resistance exercise. We will explore how hormones function as crucial drivers of muscular adaptation. By the end of this session, you will understand basic endocrinology concepts, how hormones interact, and how they target specific tissues. We will explain the vital roles of anabolic hormones and describe exactly how the endocrine system responds to a bout of resistance exercise. Ultimately, you will learn to apply these concepts to build scientifically sound training programs. Let's align our definitions. Hormones are chemical messengers synthesized, stored, and released directly into the blood by endocrine glands and other specialized cells. Endocrine glands are the specific bodily structures dedicated to releasing these chemical signals into your circulatory system. Hormones do not just float around randomly. They undergo a strict process of synthesis, storage, and regulated secretion. In the next slide, we will view Figure 4.1 to examine the body's principal endocrine organs and secondary glands that assist in hormone secretion. This diagram illustrates the layout of our endocrine landscape. Note the hypothalamus and pituitary gland in the brain, which act as the master control centers. Moving down, we see the thyroid, parathyroid, heart, adrenal glands sitting right atop the kidneys, liver, pancreas, and the gonads, testes in males and ovaries in females. Skeletal muscle is the primary target tissue we care about in exercise science. Hormones are intimately involved in regulating the balance between protein synthesis, building up muscle, and degradation, breaking down muscle. This delicate balance is driven by anabolic hormones, which promote tissue building, and catabolic hormones, which degrade cellular proteins. Hormones cannot exert an effect unless they talk to a receptor. This relies heavily on the classic lock and key theory. In this framework, the hormone acts as a highly specific key that must perfectly fit a matching receptor lock on the cell membrane or inside the cell to initiate a reaction. This illustration visualizes the lock and key concept. Notice hormone A fitting perfectly into receptor A. You can also see an allosteric binding site, where other chemicals can interact to modify the receptor's behavior. Furthermore, note the concept of cross-reactivity, where a similarly shaped hormone might partially bind to a receptor and trigger a modified response. Receptors are highly dynamic. When a hormone is overavailable or a cell wants to protect itself, it can cause downregulation, which is the inability of a hormone to interact with a receptor. Altering a receptor's sensitivity or changing the actual number of receptors can impact training adaptations, just as dramatically as increasing the amount of hormone released by a gland. Let's look at the physical categories of hormones, starting with steroid hormones. Steroids are fat-soluble, meaning they can passively diffuse directly across the lipid bilayer of cell membranes without needing a secondary messenger. Examples include testosterone acting on skeletal muscle and dihydrotestosterone acting on sex-linked tissues. This graphic tracks steroid migration. Testosterone passes through the target cell membrane, binds to its receptor, enters the nucleus, and interacts directly with DNA. This step triggers gene transcription, leading to crucial metabolic effects like increased protein synthesis, spermatogenesis, and sexual maturation. The next category consists of polypeptide hormones, which are made of amino acid chains. Classic examples include growth hormone or GH and insulin. Unlike steroids, polypeptides are not fat-soluble and cannot pass through the cell membrane. Instead, they bind to external membrane receptors and use secondary messengers like the JAK-STAT signaling pathway to transmit their signal to the nucleus. Here's the visual for polypeptide hormone signaling. You can see growth hormone binding to an external dimerized receptor on the cell surface. This activates JAK2, which phosphorylates STAT proteins. These STAT proteins from a dimer travel into the nucleus and kick off gene transcription. Third category is amine hormones. These are synthesized from single amino acids. For example, epinephrine and dopamine are built from tyrosine, while serotonin comes from tryptophan. Like polypeptides, they bind to cell membranes via secondary messengers. But a key feature is they are not regulated by negative feedback loops in the classic sense. Heavy resistance training acts as a powerful physiological stressor. Because of this stress, hormones are sharply secreted before, during, and after a workout bout. Interestingly, executing as few as 1 or 2 heavy resistance training sessions can rapidly upregulate and increase the total number of androgen receptors directly inside the trained muscle. This is a crucial takeaway. The specific force produced within activated muscle fibers directly stimulates receptor and membrane sensitivities to anabolic factors. This enhanced sensitivity to circulating hormones is what ultimately drives muscle growth and strength gains. Muscle hypertrophy is not caused by a single trigger. It is an integration of multiple complex mechanisms. Molecular signaling cascades, including those initiated by hormones, are critical to this process. Furthermore, this entire molecular landscape is augmented by neural factors that feed vital electrical signals to the skeletal muscle, further amplifying anabolic pathways. When we measure hormone concentrations in an athlete's blood, we must be careful with our conclusions. High peripheral blood concentrations do not tell us how many receptors are active or exactly what is happening inside the cell. However, we generally assume that large acute spikes in blood hormone concentration increase the statistical probability that they will bump into and interact with open receptors. What causes hormone concentrations to shift during exercise? Several physical mechanisms contribute to these blood concentration fluctuations, including natural circadian patterns, exercise-induced fluid volume shifts, changes in tissue clearance rates through the liver, venous pooling of blood in active muscles, and hormone interactions with binding proteins. Another foundational rule: hormone responses are tightly linked to the exact characteristics of the resistance exercise protocol. Your choice of exercises, volume, intensity, and rest periods dictates which hormones will spike and to what degree. Chronic long-term resistance training creates permanent structural adaptations in the endocrine system. This includes shifts in the total amount of hormones synthesized and stored, faster or slower tissue clearance rates, an increase in tissue receptor density, structural changes in a gland's secretory cells, and an increased degree of direct interaction with the cell nucleus. Let's look at our heavy hitters, starting with testosterone. Interestingly, if an athlete performs a low volume workout with very heavy loads or only one or two repetitions, serum testosterone concentrations might not change much right after the workout. However, this style of training can still expand the absolute number of testosterone binding sites and receptors. Here's the formula for men. Utilizing large muscle group exercises combined with an adequate volume of total work results in sharp, acute increases in total serum testosterone concentrations. If your goal is to design a protocol that maximizes serum testosterone, you must include these 5 specific variables. Select multi-joint, large muscle group exercises like deadlifts and squats. Utilize heavy resistance at 85 to 5% of a 1 repetition max, or 1 RM. Structure a moderate, high volume of total sets and exercises. Enforce short rest intervals of 30 seconds to 1 minute, and note that this response is significantly enhanced in athletes with 2 or more years of lifting experience. Not all testosterone is active. Free testosterone accounts for a tiny fraction, only 0.5 to 2% of total circulating testosterone. Because of this, pushing up total testosterone provides a larger absolute pool of free biologically active hormone. High volume, heavy protocols like 6 sets of 10 repetitions at 80% 1 rep max acutely raise for free testosterone in both men and women, though the absolute increase is vastly smaller in women. It is crucial to look at sex differences. Women possess about 15 to 20-fold lower concentrations of circulating testosterone compared to men. However, baseline levels vary wildly between individual women because some secrete significantly higher amounts of adrenal androgens from their adrenal cortex. Let's look at concrete study protocol. This upcoming data tracks the hormonal response to 6 sets of squat at 80% of 1 rep max with 2 minutes of rest. We'll look at both total testosterone and free testosterone responses across a timeline. On the graphs, an asterisk indicates a significant increase from baseline, and a hashtag shows a significant difference between men and women. Look at the massive difference in total testosterone scale. For men, the top purple line level spikes sharply from baseline of 32 to a peak immediately post-exercise of over 40. For women, the bottom blue line, total testosterone stays virtually flat near the bottom of the chart throughout the entire session timeline. Now look at free testosterone measured in PMOL per liter. Men experience a massive upward curve that peaks around 5 minutes post-exercise. Interestingly, look closely at the women's line at the bottom. They do show statistically significant increases indicated by the small asterisks during the workout, even though the absolute line looks low compared to men. To summarize testosterone behavior, concentration rises to meet the physical demands of an exercise protocol. Receptors will then either upregulate their binding to capitalize on that elevated signal, or they won't, depending on whether the muscle needs to rev up its metabolism. Additionally, other target sites like the nervous system or muscle satellite cells may be heavily affected during specific windows of your training cycle. Let's transition to our second primary anabolic hormone, growth hormone. While GH is famous for its role in the normal stature development of children, it remains absolutely vital in adulthood for adapting to the intense physiological stress of resistance training. This map shows the complex web of growth hormone interactions. Triggered by the hypothalamus, the anterior pituitary releases growth hormones. GH acts directly on adipose tissue to mobilize fat, on bone and on muscle to stimulate protein accretion. Crucially, it travels to the liver, stimulating the release of insulin-like growth factors, or IGFs, which feed back into muscle and bone growth. What exactly does GH do for an athlete? Its primary physiological roles include decreasing glucose utilization or sparing blood sugar, increasing protein synthesis, increasing collagen synthesis, which is crucial for structural strength, stimulating cartilage growth, enhancing immune cell function, and increasing lipolysis, which is the breakdown of body fat. Here's your design secret for growth hormone. Its release is profoundly sensitive to your rest period duration. Workouts utilizing short rest periods generate significantly greater serum GH concentrations compared to protocols that use long rest periods with the exact same amount of total work. In female populations, growth hormone baseline concentrations and acute responses to an exercise bout vary significantly based on the current menstrual phase. While we know this variation occurs, the exact underlying biochemical mechanisms are still being researched. When tracking long-term growth hormone adaptations to training, a single post-workout blood draw is often sufficient. Because growth hormone is released in pulses, concentrations must be measured over longer tracking periods of 2 to 24 hours. Scientists evaluate the total area under the time curve to determine if a training program has truly expanded an athlete's pulsatile hormone release. Now let's explore insulin-like growth factors, or IGFs, which work downstream from growth hormone. In the next slide, we will examine figure 4.7, which displays data tracking IGF-1 responses across a brutal 3-day consecutive heavy resistance training block, comparing a placebo group to a group utilizing protein carbohydrate nutritional supplementation. Look at this data trend. On day 1, both groups respond similarly. However, look at day 2 and 3. The group receiving the protein carbohydrate mass fuel supplement maintains significantly higher pre-exercise baseline levels of IGF-1, indicated by the dagger symbol, compared to the crashing placebo group. Long-term adaptations of insulin-like growth factors are complex and involve shifting transport proteins, localized muscle release, and receptor sensitivity. We cannot view them in isolation as they work alongside other anabolic hormones targeting identical muscle goals. Local tissue adaptations to heavy lifting still require deep scientific investigation. Let's pivot to the adrenal cortex and examine cortisol, our primary catabolic hormone. Cortisol exerts its main effects by converting amino acids into carbohydrates, elevating proteolytic enzymes that break down protein chains, directly inhibiting protein synthesis, and suppressing glucose-dependent functions like immune response and glycogenesis. It preferentially targets fast-twitch type 2 muscle fibers. Cortisol spikes in response to intense resistance protocols that heavily shock your anaerobic metabolism. Interestingly, acute post-workout spikes in cortisol are not necessarily negative in well-adapted athletes. Over time, the proper adaptation disinhibits cortisol's negative effects at the testicular level, allowing testosterone to maintain its primary dominion over nuclear receptors. Take note, training protocols feature high volume, large muscle groups, and short rest periods yield the highest acute serum cortisol spikes, while chronic long-term elevation of cortisol causes harmful muscle wasting. These sharp acute spikes are actually necessary to help clear out damaged proteins, facilitate muscle tissue remodeling, and preserve vital blood glucose levels. Our final major endocrine group consists of catecholamines, primarily epinephrine, norepinephrine, and dopamine secreted by the adrenal medulla. Their vital roles include increasing absolute force production through central nervous system pathways, increasing muscle contraction velocity, elevating blood pressure, increasing energy availability, increasing blood flow via vasodilation, and directly stimulating secretion rates of other anabolic hormones like testosterone. Regular heavy resistance training expands an athlete's functional capacity to secrete massive amounts of epinephrine during maximal efforts. Because epinephrine sits at the absolute intersection of metabolic control, structural force production, and downstream hormone responses, catecholamine stimulation is widely considered one of the very first endocrine responses to occur when an athlete grabs a heavy barbell. Let's wrap up by looking at a few other key chemical messengers in the body. Hormones like insulin, thyroid hormones, and beta endorphins play massive everyday roles in growth, tissue repair, pain management, and general stress coping mechanisms. For instance, beta endorphins provide crucial pain analgesia during intense training sessions. However, from a sports science research perspective, we hit a bit of a bottleneck here. Unfortunately, very few concrete data are available regarding exactly how these specific hormones respond and adapt long-term to resistance exercise protocols compared to giants like testosterone growth hormone.